My fellow scientists, my name is MengyiSun. I am a PhD student working with Professor George Yang at the University of Michigan. It is my honor today to present to you my work on a classical problem of molecular evolution, which is the relationship between translation accuracy and codon usage. Translation is one of the most important processes of cellular life. But life is never perfect. It has been estimated that, on average, one error would happen per 10,000 codons during translation. Translation errors sometimes have severe consequences. It results in misfolded proteins which might be toxic to the cell. For example, the ruthless Parkinson's disease and Alzheimer's disease are the unfortunate outcomes of failure in protein quality control. Therefore, it is important to understand how cells evolve to manage the burden of mistranslation. One hypothesis which is particularly relevant to molecular evolution is that preferred codons, or the most prevalent codons, will be translated more accurately compared to non-preferred codons. This intuitive hypothesis was first proposed by Hiroshi Akashi in 1993. In his seminal work, he not only formulated the hypothesis, but also designed a rather smart test for it. Based on the assumption that conserved sites are less tolerable to mistranslation, he calculated an odds ratio between the usage of a preferred codon in conserved sites versus non-conserved sites. The larger this value, the higher the accuracy a preferred codon is. Using this test, people have found evidence for this hypothesis in several species, mostly model organisms, such as E. coli, button yeast, worm, fly, mouse, and even human. However, it is important to stress that Akashi's test is not a direct test for the hypothesis. It relies on comparative genomics data and heavily relies on the assumption that conserved sites should have lower mistranslation rate. So far, there is no direct evidence for this hypothesis. Recently, using deep proteomics, Pupil's group from the Weizmann Institute of Science systematically quantified the mistranslation events in thousands of sites in E. coli. Their daunting effort offered the first opportunity for us to test this long-standing hypothesis directly. After reading the paper, we set out to test the hypothesis. We first cleaned the data. I'm not going to go through the details here. Long story short, we removed sites and codons whose error rates cannot be quantified due to technical difficulties. Then we merged the data for each codon, computed the absolute error rate as the fraction of wrong amino acids over the total number of amino acids mapped to a codon. Next, we normalized the error rate for a codon by the average error rate of all synonymous codons, which allows us to pull the data from different amino acids. We also downloaded E. coli coding sequences to compute codon usage measured by RSCU, the relative synonymous codon usage, which is the observed frequency of a codon divided by the average frequency of synonymous codons. Note that the relative error rate and RSCU are both normalized by the mean among synonymous codons, therefore they align well conceptually. Let's look at the results. As can be seen on the figure, the pattern looks messy among different amino acids. But in short, in more than half of amino acids, the most prevalent codon has below average error rate. However, there are also exceptions. To look at the overall trend, we pull the data from all amino acids together, and lo and behold, the relative error rate is indeed negatively correlated with the codon usage. The correlation is highly significant. This provides the first direct evidence supporting the long-standing hypothesis that preferred codons are more accurately translated, at least in E. coli. But is it true for other species on the tree of life? Well, we don't have proteomics data for other species, and yet there's one way to rescue. Remember that we mentioned Akashi's odd ratio before. Based on Akashi's logic, the larger this odd ratio, the more accurate the codon is. And it is indeed so. The odds ratio has a strong negative correlation with relative error rate in E. coli. Our result thus provide direct support that Akashi's odds ratio is a good proxy for translation accuracy. The nice thing about this odds ratio is that it can be computed from genomics data, which are abundant. This allows us to test the hypothesis for a large number of species. To this end, we leverage a recently published large-scale phylogenomics dataset. We compute the odds ratio from thousands of bacteria where we could find a closely related sister species on the tree. As can be seen here, in the vast majority of cases, the RSCU is positively correlated with the odds ratio. We obtain similar results for archaea and eukaryotic model organisms. Therefore, what is true for E. coli is true for the elephant. In general, preferred codons are translated more accurately. We did not stop here. With Akashi's odds ratio, we can address some other more subtle questions. One of the subtle questions is translation accuracy immutable across species? If it is immutable, we call this constant hypothesis. We expect a rather small variation in odds ratio across species. However, if it is variable, we expect a large variation across species, and the codon would have a log transform odds ratio larger than 0 in some species, but smaller than 0 in other species. So we plotted the log transform odds ratio for individual codons. As can be seen from the plot, there are considerable variations in the odds ratio in each and every one of the codons. We obtain similar results for archaea and eukaryotic model organisms. It is clear that codon accuracy is highly variable across species. A natural follow-up question is, what drives this variation? One idea is that there are two kinds of tRNAs that can interact with a codon. One of them is the cognate tRNA, the correct one. Another one is the near-cognate tRNA, the incorrect tRNA charged with a different amino acid, which can interact with the codon with one base pair mismatch. The competition between these two kinds of tRNAs determine the codon accuracy. If this idea is true, then tRNA composition, which changes drastically across species, would drive the codon accuracy variation. So far, evidence of this idea is limited to a small number of codons and in a small number of species. So we first validated this idea in E. coli. We compute RC over M, which is the ratio between cognate tRNA versus near cognate tRNA, representing how dominant of cognate tRNA over near cognate tRNA in competition. Indeed, the RC over M is negatively correlated with the relative error rate, no matter whether it is computed from tRNA-seq data or tRNA copy number. Note that if the y-axis is odds ratio, the correlations should be positive since odds ratio is anti-correlated with error rate, and we move to check the generality of this idea in more species. As can be seen here, this idea appears to be general across microbial species for which we can collect high-quality tRNA annotation data. In this case, the correlations between RC over N and odds ratio is overwhelmingly positive. We obtain similar results for eukaryotic model organisms. Accordingly, tRNA composition variation is likely the key driver of codon accuracy variation among species. Combining our results with previous literature, we have a model for translation evolution, which centers on the co-evolution of tRNA pool and codon usage. A key driving force is selection for translation accuracy, which is mainly determined by the balance between cognate tRNA versus near cognate tRNA. Noted that this will introduce a subtle trade-off. A cognate tRNA for one codon is the near cognate tRNA of other codons. Therefore, to reduce the error rate of one codon might be at the expense of increasing the error rates of other codons. Another important driving force is selection for translation efficiency, which is determined by the cognate tRNA concentration. Because cognate tRNA concentration affects both efficiency and accuracy, these two driving forces are linked to each other, but can also move in different directions. Finally, it goes without saying that there will be mutation and drift. In the future, it might be of interest to develop realistic mathematical models which integrate all these forces in order to understand the complex evolution dynamics of translation systems. In conclusion, the take-home message of my talks are: number one, deep proteomic data provide direct evidence that preferred codons are translated more accurately. Number two, relative accuracy among synonymous codons are highly variable among species. And finally, translation accuracy is determined by the ratio between cognate tRNA versus near cognate tRNA. At the end, I'd like to make some acknowledgements. I would like to thank my mentor, Professor George Zhang, who is the most important driving force behind the project. I would also like to thank the other members of JangLab and my dissertation committee members for their kind support and insightful comments. Last but not least, I would like to thank SMBE organizers and committee who provide me with the great opportunity to present at the Graduate Student Excellence Awards Symposium. If you have any questions or suggestions, please feel free to contact me. I am very happy to chat about it.